Welcome, everyone. So this is actually the second network seminar of the third year. So this has been going on for some time. Véronique is actually organizing it, this series. She's doing a great job. So today we have people from BC2C, or the BC Cancer Consortium. And of course, as usual, we have some members of the patient working group who are there. I will let Véronique introduce Dan Murphy. And of course, we have people from the network, clinicians, scientists, trainees, and of course, we hope, I think also people from the Terry Fox Foundation. So it's very nice for everybody to be here. And I think we have a packed agenda, I'm being told, so I shouldn't talk too much. So there you go. It's your turn, Véronique.
Merci beaucoup, André. Welcome, everyone. I will start with a little bit of housekeeping. So as just a quick reminder, this is only our second event using the Teams platform, so it's a little bit new for everyone. So I just want to say right off the bat, if you have questions for the speakers, we're going to keep questions for the end of the seminar, but you can use the Q&A function, which should be at the top of your screen. Right next to the chat, there's little speech bubbles and that say Q&A underneath. So you can type your questions in there, or once we get to the question session, you can raise your hand and then we'll allow you to unmute. So I'd also like to take a quick moment to acknowledge that for me personally, I'm calling in today from Mi'kma'ki, the traditional and unceded territory of the Mi'kmaq people. Our network members and our speakers and all of our attendees today are, of course, located throughout the country or on lands that are home to diverse First Nations, Métis, and Inuit populations. And I'd just like to invite everyone to think about that and recognize those Indigenous peoples and reflect on the importance of those lands where you are. And so with that, I would also like to introduce my co-chair, Dan Murphy. Dan joined the network's patient working group almost a year ago now, and he's an integral member of the group. On top of all of the work that he is doing and the whole working group is doing, Dan generously volunteered to participate in today's event. So thank you so much, Dan, for joining us. And I'll pass it to you if you want to tell us a little bit about yourself and get this talk started.
Sure. Thank you, Véronique. I'm really happy to be included today. My wife, Kathy, and I live in Toronto with our 2 university student daughters. Sadly, in June of 2023, our lives were upended when I was diagnosed with stage 4 pancreatic cancer. 2 years to this week that I started chemotherapy, as a matter of fact. These last 2 years have certainly been difficult times for us, but in many ways, they have also been remarkable. Remarkable because despite the terrible prognosis that I and the vast majority of pancreatic cancer patients face, I am living with cancer actively, feeling good, and experiencing great quality in life. I owe this in large part to precision oncology and the positive response that I've had to the treatments from it. Last fall, as Véronique mentioned, I joined the MOHCCN patient working group so that I could play a small role in creating hope. Hope that more patients across our great country can have access to treatments like I've had, and hope for their improved quality of life, and hope for continued advancements to benefit generations of future patients. It's both an honor and incredibly fulfilling to be a part of this. With that, I'd like to introduce our first presenter. Dr. Daniel Renouf is a medical oncologist at BC Cancer. Dr. Renouf is one of the British Columbia Cancer Consortium leads, and today will be giving us an overview of recent activities within the consortium. Over to you, Dr. Renouf.
Thanks so much for that kind introduction, Dan, and the offer to present today, and for our BC Consortium to present Véronique and Andre. Véronique, would you mind pulling up my slides? Great to have, I see we have over 60 people on, so great turnout. And I'm going to just give a few opening remarks on behalf of the leads of the BC Cancer Consortium, and then hand it over to my colleague, Dr. Carsan, to kind of dig deep into some science and exciting work that his group's been doing. Next slide, please. So I'm coming today from beautiful sunny Vancouver. In Vancouver and specifically at BC Cancer, we're on the unceded traditional territories of the Musqueam, Squamish, and Tsleil-Waututh nations, and very thankful to be able to work and live on these lands. I think we're very blessed. And as Véronique said, I know we were joining from across the country, so please take a moment to acknowledge the lands that you're joining from. Next slide. Just a bit of, I think you skipped a couple, Véronique. Yeah, okay, yeah, this is good. A little bit of background on BC Cancer Consortium. We are one of the founding members of the Marathon of Hope network as of, I guess, 2021. We, this presentation and overview behalf of a huge team of people. I think I see quite a number of them on the seminar today. So a huge thank you to them, and a huge thank you to really extensive funding support that we've had to support kind of the match funding for this, including BC Cancer Research Institute, our fantastic foundation here, BC Cancer Foundation. A lot of work done in part of the pediatrics group, so the BC Children's Hospital Research Institute and Children's Hospital Foundation. We've got extensive support from Genome BC, and very thankful for that, as well as from the UBC province of British Columbia, and then quite a number of funders supporting each of the individual cohorts. So really a huge thank you, and none of this could be possible without many, many funders coming together. And I always think this is one of the amazing things about this, and when Victor Ling first conceptualized this, how can we bring quite a number of funding committees and people, galvanize them together with one vision? And I think just thinking that, over 20 something different foundations and funding committees are coming together to support this is a good example of that galvanization. So that itself represents a huge success of this network. And then a great deal of support from an infrastructure perspective from Michael Smith Genome Sciences Center and multiple CFIs and through CGen. And that reminds me that my co-leads, I don't know if any of them are also able to join. I know Marco is traveling today, but huge thank you to them for all the support in leading this initiative. Next slide, please, Véronique. So I think at BC we've been quite successful. We've sequenced quite a number of cases. We've really focused on 7 cohorts: AML, which you're going to hear more about today, breast, lymphoma. I think we've had past seminars on some of these areas: breast, lymphoma, pancreatic cancer. We have sarcoma. We have pediatrics. We have prostate cancer, and then our POG program, which reaches into many of these disease sites. And we also have a CLL and another breast cohort joining as of 2025 to 2026. We've sequenced quite a number of retrospective cases, but also have a huge focus on prospective cases. And the Personalized Oncogenomics Program, or POG, has really been the framework for doing that. And in my own personal research, we've done quite a number of cases in pancreatic cancer, and so what Dan says really resonates with me is, how can we use this information to not only guide science, but guide treatment decision-making for patients going through cancer in real time? And I think one of the really exciting features that many of you know is that's really going to be a focus of this network moving forward is prospective nature. And I'm going to highlight over the next couple slides some areas that we've learned and are very welcome to kind of share our learnings with the community around kind of some of the prospective, I guess, challenges and solutions that we found. Next slide, please, Véronique. So one of these is, I guess, data sharing. This represents both retrospective and prospective cases and is important. But obviously, this is a key element of Marathon of Hope. And a lot of infrastructure is required. And we've had some good success in data sharing with a couple of the consortia. One regarding multiple myeloma, one regarding breast cancer profiling. And again, I think these type of infrastructure initiatives are key elements as we move forward. And we can learn from each other across the network. And a lot of the, I guess, pipeline created an infrastructure for POG or personalized oncogenomics can be utilized to, doesn't necessarily have to be reinvented or redeveloped. This program has been going on for, I think, a decade now. So it's we've had many iterations of it and learned a ton. I think these are two examples, but many others of where we can deploy that to support the network moving forward. Next slide. This is another example. This is around PORI, which is our platform for oncogenomic reporting and interpretation. Reporting and interpretation. And again, this was created quite painstakingly and many iterations that has been published as a means of how do we take all of this information and then generate kind of an actionable report that can then be utilized to make treatment decision making in real time. And that's really the key, I would say, challenge and opportunity power of doing prospective analysis is utilizing this information to make real-time treatment decision making. And so that's, again, I think we've had a collaboration with the Prairie Consortium, UFC, in this regard, and definitely I think this is another key area that can be deployed across the greater network as we move forward. Next slide. So again, what can we do to support prospective cases? We're committed to help set up the elements needed for prospective return of results. We have quite a number of SOPs that have been developed and redeveloped and worked on over the years around protocols, ethics approvals, how we access samples. We're doing a lot of work around FFPE versus fresh sample collections, lab SOPs, project SOPs, etc. So this type of work is extremely important, I think, infrastructure as we move towards more prospective cases moving forward. Next slide. So to summarize, I think we're very excited to be part of this network. I'm really excited about the future steps that the network is undertaking. Amazing science has been generated from a lot of the retrospective analysis, but we really want to focus on moving forward, prospective cases and how we can influence real-time treatment decision making. And I think there's a lot of lessons learned that we're quite keen to share as we do that. We're aiming to ramp up our sequencing over this last year and again, really looking at how we can support real-time impacts to improve patient care using precision guided genomics and precision medicine. So with that, I'll stop and want to make sure we have ample time to hear some exciting science from my colleague, Dr. Carson. So hand it back over to you, Véronique and Dan. Happy to take any questions in the chat, or feel free to reach out over email if you have any questions or follow-up or collaborations that you're interested in after the fact. Thanks, everyone.
Thank you so much, Dr. Renouf. We are running into an issue with Ali being able, or Dr. Carson being able to actually join the meeting. So Derek, could I ask you to do your presentation now, and then we'll try to get to Dr. Carson after that? Okay, yeah, great, absolutely. Thank you. So Derek Tam is a bioinformatician in Dr. Carson's lab, and he also did his master's degree in Dr. Carson's lab. And today he'll be telling us about, I think, some of his master's work, tracking clonal structures in AML. So over to you, Derek. Thank you.
Perfect. Thank you for the introduction, Véronique. I appreciate the opportunity to talk at the MOHCCN meeting. So yeah, I'm Derek. I am a recent graduate of the Bioinformatics graduate program, where I got my master's from the University of British Columbia. And yeah, today I'm going to be sharing a little bit of the work from my thesis and a lot of the things that we've been working on, following up on that since. So my intro is light because I was depending on Ali giving a little bit of background, which we had coordinated, but I'll just jump into it. So acute myeloid leukemia is it's a type of leukemia that is highly heterogeneous. So what that means is, in terms of the mutations, there's a low mutation load, but we still see a complicated clonal structure. So AMLs are seen to transform and acquire new mutations, and then these clones can branch and expand in response to various selective pressures like therapy, and then that can result in great diversity of clones. So this is important because part of understanding therapy resistance and why the relapse rate is so high in acute myeloid leukemia will be built on being able to understand clonal structure in a high resolution and in an accurate way. So this is a bioinformatic problem that many people have been working on for a very long time. Originally, methods revolved around bulk sequencing. So a lot of the clonal proportions would be inferred based on allele frequencies. But now with the advent of single cell sequencing, we can actually more directly identify which mutations are co-occurring. But with single cell, you still have a lot of technical issues like noise and dropout. But whichever method you use, fundamentally, these approaches really rely on gene mutations. So really the clonal structure that you will infer reflect the cancer's genetic landscape. So more recently, additional methods have been proposed, specifically this one in 2019, where basically clonal architectures are being inferred using mitochondrial DNA mutations. So a very quick primer on mitochondrial DNA. I think even people who are in genomics probably don't look at mitochondrial DNA that often. But mitochondrial DNA has a much higher mutation rate relative to nuclear DNA. And this is in part due to the replication machinery and generally being exposed to oxidative damage. So mitochondrial DNA, like nuclear DNA, are heritable. So as a cell divides, the mitochondria within are subdivided into the two daughter cells. So any mutations that those mitochondria carry as they divide will be inherited by those cells. But most importantly, mitochondrial DNA mutations actually occur independently of nuclear DNA mutations. So they're not directly correlated or related in any way. And mitochondrial DNA mutations don't typically affect clone fitness or proliferation at low frequencies. So that's all well and good, but how do you apply it to actually infer clonal structure? So the way it's being used is kind of like a barcode or a marker. So the cells, as you can see here in this little cartoon, as they divide, the mitochondria within are subdivided. And if you look at the specific mitochondria, you can see each cell's got multiple mitochondria, and each mitochondria's got multiple copies of mitochondrial DNA. And in this particular copy, you've got this mutation. So as that cell divides, that mutation will be inherited by the two daughter cells. So regardless of the specific heteroplasmy, depending on how much sequencing depth you have, the fact that you can detect this mutation in these two cells tells you that these two cells must be more closely related to each other than they are to the other two cells who don't have the mutation, essentially. So if you scale this up to many thousands of cells with many more mitochondrial mutations, you can imagine how this method could give you a fairly detailed clonal structure. So our goals here is basically we want to develop a method that will use this mitochondrial variant technique in combination with mutational profiling. And then we want to apply this method to longitudinally paired samples from refractory and relapsed AMLs so that we can basically understand how clonal structure is changing between those time points. So the samples we obtained are from the AML patient cohort from the Marathon of Hope. And we basically included patients that were refractory to treatment, or they initially responded and then relapsed. But importantly, were not transplanted since the addition of external clones would be a confounding factor. So these paired diagnosis and relapse time point samples were then assayed using a custom targeted single-cell DNA sequencing panel. So the panel is basically, it's comprised of two parts. So one part is a myeloid panel. So this targets genes of known interest in myeloid malignancy. And then the second part of the panel is this custom mitochondrial DNA panel that basically tries to cover as much of the mitochondrial genome as possible so that we can catch those random variants as they occur periodically. So we ran it on all of the patients you see here, and each sample is represented from the diagnostic time point and the relapse time point. So every patient is represented twice here, and they're annotated at the top by the time point of that sample. So what you're looking at is basically a very high-level summary of the genes that are mutated in each patient. And the patients are actually clustered from left to right based on the mutations that were identified. And it's actually, essentially, if we cluster them based on this broad spectrum of myeloid mutations, we don't really see a pattern that gives us a way of predicting whether or not a patient will relapse, or if there's a pattern specific to relapse that you don't see in diagnosis. But obviously, this is a single-cell technique, and this is a very generalized summary. So we can dive into a specific case here. So we're going to look at AML24 DNR, and we're going to follow this case so that you can see basically how the analysis worked. So what you see here is a summary of basically every mutation that we captured in every cell in AML24. In each row, you can see a locus and the specific mutation that was present. And then in each column, grouped together, you have basically the cell proportions. So the wider a column is, the more cells are present in that group. So I want to bring your eyes to this row here, where you can see the cluster annotation. And this basically identifies cells that carry the same mutation. So for example, this dark brown cluster here, it's homozygous for these two mutations, and that's why they're in this brown cluster. And then if you look at this light brown cluster here, you can see because they're wider, these clones are more abundant. But importantly, they have a different mutational profile than that other clone. They're only homozygous for this first one, right? And then finally, if you look at this green one here, you can see this is a very small clone that was detected at diagnosis, and you can see it expanded a little bit at relapse, right? So this visualization is not very conducive to understanding how the clones change over time. So what we can actually do is represent all the same data as a fish plot. So these are the same cells, but what you have now is from left to right, a progression of time, and you can see the sample captured at diagnosis and the sample captured at relapse. And the vertical proportion tells you how abundant that clone was at that time point. So if you look at the sample here, you can see back at that, that light brown clone, you can see it's abundant at diagnosis, similarly abundant at relapse. And then if we look at that green clone again, you can see, it's barely detectable at diagnosis, and you can see that expanded a little bit at relapse. So if we do this for all of the patients, you can see that the relapse structures actually seem to occur in a spectrum. So there's these different patterns that you can kind of stratify the cases on based on the relative amount of change in clonal structure that you're observing. So on the left here, you can see that case we just looked at, and two others where the apparent clonal structure is has either very little change or no change. And the same clones that you see at diagnosis are present at relapse in similar proportions. This is in contrast to the cases you see on the right. So at the top, you can see the clones that are detected at relapse are, in terms of mutations, not even detected at diagnosis. So these were clones that emerged and evolved since treatment. And then in AML1, you can see that the dominant clone at diagnosis is barely detectable at relapse, right? And then you get cases that are kind of in between. So the cases on the right, they're kind of straightforward in terms of interpretation, right? So the variant that was present somehow modified the clone's fitness and in response to that treatment, and that caused that clone to contract or a different clone to expand. But more interestingly, the cases on the left, because the mutational landscape is the same, we don't actually understand why the relapse occurred, because technically no new mutations were occurred in any kind of substantial way, but relapse did occur. So if we look at these cases, part of understanding why that happens is understanding the model and the data that was given. So in this specific case, the data that's used to infer the clonal structures is based on nuclear DNA. So this kind of, any change that you see will be reflective of the clone fitness as it's correlated with mutational differences. So if you don't see a clear change in clonal structure, then it means that the mutational differences are not sufficient to basically tell the whole story. So the model's incomplete. But because we captured the mitochondrial variants, we can actually see that if we took the same cells, except we looked at the mitochondrial variants and we reclustered them, on the right here, you see the same cells regrouped into new clones based on mitochondrial variants, and then it becomes very apparent that there is a clone that was detected at a very low frequency at diagnosis that expanded to basically repopulate the entire relapse. So if you look at the bottom here, this is the actual data that went into it. And there's 283 mitochondrial variants that were identified to have these very subtle variations between those cells. And this was the information that was used to infer those clones. So if you compare these, it kind of looks like if you look at the myeloid mutations, the clones had resisted treatment, so no clear change had occurred. But if you look at these stochastic mitochondrial mutations, it looks like the clones did respond to treatment. But if you look at some other cases, you actually see the opposite. So in that case that we looked at earlier, 103, you see that the pink clone that was dominant at diagnosis, that was undetectable at relapse. But if you look at the mitochondrial variant breakdown, those clones appear unchanged and the clones look like they resisted treatment. So we basically wanted to quantify this change and then see what the pattern in the relationship was. So we calculated how much divergence had occurred between diagnosis and relapse. I'm so sorry, I think they're testing the fire alarm in my building. So then each point here represents a patient, and its position on the x-axis basically tells you how much change had occurred structurally from diagnosis to relapse as captured by the myeloid mutations. And then on the y-axis, it's the same thing, but for the mitochondrial variants. So if basically when you see a change in the clonal structure based on myeloid mutations, and you saw the same change in the mitochondrial mutations, then you would expect a lot of the samples to fall along this diagonal. But obviously, that's not what we see. So interestingly, in some cases where myeloid mutations do not capture the difference in clonal structure, we can see that mitochondrial DNA mutations actually reveal that some kind of clonal selection is occurring in the absence of a correlation with myeloid mutations. On the inverse, there are cases where there is a clear change in clonal structure that can be attributed to myeloid mutations, but mitochondrial DNA mutations actually don't capture any additional subclonal change. So what we can infer from this is basically that the clonal structure based on mitochondrial DNA mutations reflects some kind of biological difference that is not captured by myeloid mutations. And this biological difference is a factor in clone fitness. So basically, what we're capturing with the myeloid mutations is not the same as what we're capturing with the mitochondrial DNA mutations, and both of them are playing a part in response. And furthermore, if we look at the refractory cases, so we stratify these cases by their type of response, we can actually see that almost all of the refractory cases have very little clonal structure change, and this is independent of any myeloid mutations. If any mitochondrial DNA mutations were capturing kind of like non-genetic differences, then this result actually suggests that refractoriness is correlated with non-genetic changes, and that refractoriness is occurring prior to treatment. And what we're seeing in cases that respond and then relapse is actually the same pattern. Okay, so in summary, of the 23 cases, 75% of them seem to have no clear change in the mutational landscape. But in approximately 1 in 3 cases, we do see some kind of clonal change when we use mitochondrial mutations that is reflecting a difference in clone fitness. So if we were to follow up, these are really the cases that would be quite interesting because whatever difference biologically is present between these cells, in this example, this green clone and this red clone, that's causing the green clone to respond to treatment, or to resist treatment, but the red one to respond, that is not genetic, would be very interesting to find out. So in conclusion, we designed a novel single-cell DNA sequencing panel, and this was able to recapitulate clonal structure. We saw that this change kind of occurred on a spectrum. And of the cases with very little structural change, we saw that in approximately 1 in 3, there was some kind of subclonal change that was detectable with mitochondrial DNA mutations. So kind of the final novel hypothesis from all this is that if the mitochondrial DNA mutations are capturing some kind of heritable non-genetic difference, then this biological difference is a factor in clone fitness. So bringing it all home, what is the potential impact of this work? So clone diversity in AML, as we know, is a contributor to refractoriness, and it plays a part in the high relapse rate that we see. So being able to understand all the factors that are contributing to clone diversity will allow us to identify new ways of addressing each of these factors, which will hopefully improve treatment effectiveness and reduce rates of relapse, and then ultimately, hopefully provide better outcomes for patients. So a lot of people to thank. I want to thank Ali and Dr. Andrew Roth, who were both supervising me, and then many people in my lab who contributed to this work or guided me in some way. In particular, I want to call out Christy here. Christy is the new graduate student who's basically picking up the work where I've left it. Yeah, so thank you for listening.
Thank you so much, Derek, and great job continuing on with that test in the background. The good news is that the audio screening worked pretty well, and we couldn't hear very much of it. So great talk. Thank you. I see that we had Dr. Carson back, but we've lost him again. So I'm thinking maybe we can open it up for questions, and if he can make it back, we'll fit in his talk before the end of the hour. So maybe Derek, if you could stop sharing your screen. And then if anyone has any questions, as we said in the beginning, you could put them in the Q&A. This is the little speech bubbles at the top of your screen on the left. You can also raise your hand using the little hand button that's, I think, 2 over from that, 2 to the right from the Q&A. And if you do that, we can unmute you and allow you to ask your question live. And I think, Dan, you might have a question to kick us off.
Yeah, for sure. Thanks. So, Derek, as a patient, your passion and curiosity about this is really inspiring, I have to say, to start it off. But I know you touched on near the end of your presentation, you mentioned if you were going to look into something next, you were going to look into that one cohort. Is there anything else that surprised you in all the work that you've done as something that you really think that you'd be inclined to dive into to next? You can elaborate on that maybe, but thanks.
Yeah, thank you for the question, Dan. Yeah, so I think I highlighted that small subgroup where we would expect to see something, but really like the other 2/3 of those patients where we don't see anything with like the myeloid mutations or with the possibly epigenetic differences between these cells, that would be like a huge black box. Like we don't understand why those cases are relapsing at all then. And I think that would be a very interesting place to follow up. Yeah.
Thanks, Derek. We have a question in the chat from Amber. I think it's sort of along the same lines of what are the next steps and future plans for this project?
Yeah, so we've made some pretty good headway into the next step. So of those cases, we've done additional assaying basically of the epigenetics of those same libraries. And one of the cool things about using mitochondrial mutations is that if you have a different sample, so different cells from the same patient, and you've assayed them in some way where you can also capture the mitochondrial variants there, because that fingerprint is present in all the cells in that person, you can actually pair data across assays, even if they're not literally the same cells. You can kind of look at the same clones. So we're basically trying to do that to answer that question where we want to know what's happening in a very directed way.
All right, thank you. Sherry, I see that you have your hand up. You should now be able to unmute yourself.
Yep, I can. Thanks. Thanks for the great talk. I don't know if this is addressable, this question, or if it's very naive, but what do you think biologically is happening that the mitochondrial mutations are causing these cells to become resistant?
Yeah, so our kind of main hypothesis is since it's not genetic, it must be epigenetic. I hesitate to say that because I currently have no proof that I can share. And I also hesitate to say it at my defense because I didn't want to get grilled. But we think that's what it is. So in some of the other work in our lab, we've shown that differences in methylation, which reflect differences in cell type, are actually predictive of responsiveness to treatment. So one of our leading thoughts is that it's actually the epigenetic state, which is set prior to possibly some of these mutations being acquired, that is happening earlier, that's kind of defining the responsiveness of these cells. So sometimes the mutations play a part, but we think that in those other times when it's not, it's probably something to do with the epigenetics.
Thanks.
Great, thanks for that. Dr. Nielsen, you should be able to unmute yourself and ask your question.
Okay, yeah, hi, thanks. Really interesting talk. Maybe it's premature to ask this, but these epigenetic changes are more plastic and quicker to maybe be selected for or against than DNA mutations. What's your sense, in terms of drug resistance and acquired drug resistance? Do you think this might be a predominant pathway that's, really the way cancer cells are most likely to develop a resistance on the fly to treatments we give, as opposed to waiting for stochastic mutations and clonal selection of those?
Yeah, thank you for the question. Yeah, so based on what we've seen in some of the other projects in Ali's lab, we do think epigenetics plays a very large role in predicting responsiveness. But we think those factors are very interactive. So like you said, the epigenetic landscape can be quite plastic. But in some ways, we don't really know the order of events. So it could be, like in AMLs, we know that there's like early mutations that typically affect like epigenetic readers and writers. So if those are they're characteristically early mutations, it could be that that's because the epigenetic state needs to be shifted in some specific way for the cancer to then like further progress, or that it's something that we just see more frequently. So there must be some kind of fitness advantage to doing that. So we don't know exactly what the order of events is, and that's part of like deconvoluting the data that I have too, since I basically have two different types of clustering for the same time point, and they're not necessarily always perfectly consensual. So that could mean that the data is wrong, or it could mean that the order of events is not exactly always, epigenetics first, then genetic mutations later. So I think that's I think it's part of the answer to like why relapse and resistance is occurring, but it's a complicated problem. Yeah.
Thanks for that. I'm seeing another question in the Q&A. So Rosaline asks, if these changes are epigenetic, that would mean that they are reversible, right? Could then we hope that those patients experiencing mostly mitochondrial DNA mutations could again respond to prior treatments?
Yeah, so I think that's that's definitely, I think, a good avenue of treatment. And I think there are some drugs in clinical trials where essentially the idea is to shift the epigenetic state of a cell that looks resistant to an epigenetic state that would be sensitive. And then in combination with a therapy that works on the sensitive cells, you could basically induce sensitivity and then basically target more of the leukemic stem cells. But yeah, it's definitely something that's being explored.
Great, thank you. Unfortunately, it doesn't seem like Ali's been able to join again. Are there any other questions either for Derek or for Dr. Renouf on the BC Cancer Consortium more broadly? I think so, unless someone is typing in the chat, but I don't know. Derek, are you familiar with Ali's slides at all?
I don't have the confidence to present them. I think both of us have been revising quite thoroughly since we last met on Monday, but
yeah.
Yeah.
Okay, well, that's really unfortunate that we weren't able to make that work. I suppose if anyone is able to stay, we can hang on in case, but oh, Jess, I see you. Your hand up. Jess, you should be able to unmute yourself.
Oh, there we go. We're trying to send someone over to Ali's office. He's on site, so we're hoping we can get some systems over there. I don't know how long that will take. I recognize we only have 13 minutes left.
Yeah.
So.
Thank you for trying. We can. Any other questions? Maybe? Okay, well, unless Jess, you have noticed that Ali can join us imminently, I think maybe we'll wrap it up here.
He just sent an email saying it's not going to work.
Yeah, okay.
Sorry.
Well, that's unfortunate, but thank you so much to Derek and to Dr. Renouf for the talks. It was great to learn more about research going on in BC. And thank you for everyone who attended. We had a really great turnout today, so we really appreciate everyone joining us. And we'll see you probably in the fall, but you'll hear from us about the next seminar.